So in last lecture we have gone through the cell wall, and in today's lecture we are going to look for the other features or structure of the typical bacterial cell. So we are looking for the prokaryotic cell, and we know that prokaryotic cells includes mostly bacteria, or bacterial cell is the best example of prokaryotic cell. So we have gone through the cell wall of bacteria, and in today's lecture we are going to look for the cell membrane. Now the cell membrane is the innermost part of the cell, which is located just below the or just behind the cell wall, and it is protective in nature. Cell membrane is made from phospholipids, proteins and carbohydrates, forming a fluid mosaic type of structure. It surrounds bacteria, and it is most important in the bacterial cell. It controls the movement of substance in and out of the cell. So say for example some molecule of water or some structures is required to be transported towards the inner side of the cell. This cell membrane will occupy that particular or perform that particular function of the transport. So transportation is controlled by this particular type of membrane that we call it as a cell membrane. Now here you can see we have discussed in last lecture, the outer membrane is there, that we called it as a cell wall, or sometimes this outer membrane is also known as the capsule, followed by the peptidoglycan layer, and this peptidoglycan layer is followed by the cell membrane. So the most internal part you can see, that is nothing but the phospholipids, and peptidoglycan layer is there. Now in the second diagram we can see that the cell wall is having the next part that we called it as a cell membrane. So here the cell membrane is there, that is made up of phospholipids bilayer of the membrane. So here the yellow color structure is nothing but the phospholipids bilayer, and blue color structure you can see that we called it as an integral protein. This integral protein is the most important protein for the transport of molecule from the outside of the cell or from the inner side of the cell. So this is nothing but the structure of the cell membrane. Here the structure of phospholipid is given. So bilayer of the phospholipid is shown in this particular diagram. Now moving to the next, that is the other layers which is present on the external side of the bacterial cell, or it acts as a protective covering for the bacterial cell. So here we can see that capsule, slime layer or S layer, these are the different layers which is present in bacteria and work as a protective layer. Now some bacteria have a layer of material lying outside of the cell wall. When the layer is well organized and not easily washed off, it is called capsule. Now a slime layer is zone of diffuse unorganized material that is removed easily. The next one is in a gram positive bacteria and gram negative bacteria have regularly structure called S layer or of their surface. S layer has a pattern sometimes like a flower, like as it is accomplished of protein or glycoprotein, and it layer, that S layer, is a protective layer, and it will help in a protection of that particular organism, and it will provide rigidity to the cell membrane, rigidity to the bacterial cell, and that's we can say most important layer which is present in the bacteria. So different types of layers are present on the external side. So this capsule, slime layer, S layer, these are the layers which is present on the external side of the cell. Now the next one is pili or fimri. Now as we know that this bacteria or bacterial cell is required to be attached to something. Say for example the bacteria required to attach to the substratum, or it is required to attach with the other bacterial cell. So there is a different types of organs which is developed in a bacteria, particularly pili and fimri. These are the two types of organs which is developed in bacteria that helps this bacteria for the attachment to the different parameter. Many gram-negative bacteria have short, fine hair-like appendages that are thinner than flagella and not involved in motility. These are called fimri. Pilus are similar appendages about 1 to 10 per cell that differ from fimri in the following way. The one, pili often are larger than fimri. Next, they are generally determined by sex factor or conjugative plasmids and are required for the bacterial mating. And then last difference is there. Some bacterial viruses attach specifically to the receptacles on sex pili at the start of their reproductive cycle. So we can say that this pili and fimri act as a appendage, or we can say organ, for the attachment of this particular bacterial cell. So pileus is a similar appendages about 1 to 10 per cell and differ from the fimri. So these are the two different we can say organs, fimri and pili. So they work for the different parameters attachment, and one is act as a sex determination, or it will helps in a reproductive cycle. Now the next thing is there, that is flagella and motility. As we know that bacteria is a most smaller, or we can say it is a microscopic structure, but though they are microscopic, some bacteria shows the motility. They will move from one place to another place, particularly in a liquid state, or we can say liquid medium. They will move from one place to another place. So in this particular work, they use this flagella, or they use the flagella like structure for the motility. Motile bacteria have moved by the use of flagella. Flagella is thread-like locomotor appendages extending outward from the plasma membrane and cell wall. So cell wall and cell membrane, from this the flagella is produced, or it is developed. They are slender in shape, they are rigid structure, and it is having about 20 nanometer across and up to 15 to 20 meter long type of structure is observed, and that we called it as a flagella. So need to remember that flagella is useful for the motility, pili and fimri is useful for the attachment. So that is the basic difference between pili and the fimri and the flagella. Now here you can see, in some bacteria only single flagella is observed. In some bacteria two flagella is observed. In some bacteria on the both sides two different types of flagella is observed, and in some bacteria more than 5 or more than 10 the flagella is present, which covers the total body of that particular bacteria. So depending on this type of bacteria, the name of this particular bacteria will change. So here you can see monotrichous means having the single, amphilotrichous having the two flagella on two different sides, then tetrapolyno. So these are nothing but the different types of flagella. Flagella is a hair-like structure. Flagellar filament are made up of subunits of single protein flagellae. The transmission electron microscope studies have shown that flagellum is composed of three parts: filament extended from the cell structure, basal body embedded in the cell, and hook-like structure which is present for the attachment of filament and basal section. So here we stop with this discussion that pili, fimri and flagella. So watch this video 2 times to understand the concept of locomotory and non-locomotory organs in bacteria.